Hello, hello, hello there, students. Welcome to the fourth week of our course, Biology 1406, Intro to Biology for Science Majors, where this week we're going over cells, cell structure to be specific. So let's jump right into it. Um, what are cells? And the textbook term is that just cells are the fundamental building blocks of all living organisms. It's the worst line ever made, but you can still see. The operative word being living. Um, and what do I mean by this? So when we talked about our first lecture, when we talked in our first lecture, one of the criteria for life is that um, it was anything that was comprised of one or more cells. So this has like this sort of circular reasoning to it, where life is cells, cells have life, but really cells are or anything that is qualified as living has at least one cell. This could be something simple like bacteria or something complex like us. Okay? Humans. Every organism, every system, every organ, every brain tissue, everything has a cell. Okay? And those cells share common characteristics, but they also have great diversity. So, what are we going to talk about today? We're talking about cells, yes, but this is what we're going to break down as far as your chapter goes, your reading goes. How do we study cells? You may already know the answer to this, but I'm going to go into a little bit of detail on that. We're going to talk about prokaryotic cells and eukaryotic cells and what this means. The difference is when we talk about prokaryotic cells or, bacterial cells versus the more complex eukaryotic cells, which, humans, plants, fungi, stuff like that. And then we can just link all this, the endomembrane system, the cytoskeleton, whoop, all that into a term called organelles. Okay. And that specifically will be about eukaryotic cells, but there are some prokaryotic cell attributes we'll talk about too. So cells are the building blocks of life. And the analogy we have here is we have a brick wall. And if you look at a brick wall and it's macro, it is a giant wall. But the individual units that build it up are just bricks. So if we look at this, we could kind of put another analogy to this when we talked about macromolecules and monomers and putting monomers together, you got polymers, right? Monomers became polymers. So you had the building block and then you had the structure. Okay? Building block, structure. The building block amino acids come together to make proteins, right? And nucleic acids come together to make DNA and RNA. Okay? So cells are the building blocks of life. It's how I try to remember it. But every cell, even though you have this idea of what a cell is, there's a lot of diversity to it. Um, you have bacterial cells for starters, but if you grow and you get to more complex systems, you have epithelial cells, which is a eukaryotic cell. That's like your skin. Bone cells for structure. You have immune system cells like white blood cell. You have blood cells that are red blood cells. Um, there's an enormous amount of variety. So how do we see all this stuff? Now, again, we have our eyes. Part of science, part of observation was that we need to be able to see the thing we are studying. Until then, it does not qualify as science. So if you cannot observe it, it doesn't fall into that sphere of science. It doesn't mean it won't one day. Think of the Galileo and the telescope. He had a device, he had a scientific breakthrough, a new piece of technology, but he really only had access to it, which is the kickback he got from his discovery. So, how do we study cells? We study cells through this process called microscopy, which, if you just break that word down, I'm sure you all can get where that's coming from or get where we're going. It's just a microscope. And we cannot see individual cells. We need a microscope. And until the invention of it, we didn't know what was happening. We didn't know about cell theory, which we'll talk about cell theory here in a second. Um, so yeah, we're looking at microscopy. We're looking at light microscopes, which are the ones you're most common with, is which is what this microscope up here that you're seeing. Essentially, what you do is you put an object on this platform. Light comes through. In the case of modern telescope or microscopes, you have an electrical connection. You plug it into a wall, a bulb shines through. Back in the old days, you had a mirror. The sun would help you shine through, or a candle. But yeah, light shines through, lights up the object, and these little guys right here are called objectives, and they allow you to magnify and see the device on that platform. And you can get up to 400 times the size of the object. So this is handy. You can actually see human cells. As long as they're stained, um, we use something called methylene blue. And you can actually see like the cell membrane and the nucleus with a light microscope, which if you take my course and you get to play with microscopes and see that, if you take it in person, you will not get that luxury if this is if you're taking this online. But yeah, one day maybe you will be able to. Now, unfortunately or fortunately, or however you want to look at this, just kind of the nature of things, there are things that are much too small for light microscopes. And this is where we talk about these things called electron microscopes. Now, if you remember in that first week where I introduced myself, the What is Science lecture, I showed you those slides on pollen that I took. Well, I took those with what's called a scanning electron microscope, SEM. Now, the idea behind a scanning electron microscope is, let's say this is our object right here. Basically, you have this electron gun that fires electrons, and basically the electrons will ricochet off in a certain pattern, and the computer measures that angle of dispersion, and it results in an image. Okay, and that image is what you see. And where a light microscope can only get you 400 times the magnification, SEMs are really nice. Scanning electron microscopes can get you anywhere from 3 to 5,000 times. Okay. Those pollen pictures I took were about 4,000 times the size of what you would expect to see or 4,000 times magnification. You can go even further to something called a transmission electron microscope, and this you can see up to 100,000 times with these. The same concept, firing electrons. I know with a scanning electron microscope, you coat the object in gold. That's how it allows the electron to bounce off. Very expensive piece of equipment, but I was fortunate enough to be able to use one. Um, so yeah, so all this to say is we took all this stuff, and again, um, we have this thing called the unified cell theory. Now, how do we come about this? So you always have to remember when we're studying history, especially scientific history, the science you know it as today is not the science they had back then. We're privileged with having a microscope, with having these advances, this medical advances, understanding what cells are, and you're just like receiving this information. But there was a time we didn't know any of this stuff. We didn't know what a cell was because we didn't have a microscope, which is where Antony von Leeuwenhoek kind of came in. And I don't ever expect you to remember names, but basically he had some handcrafted lenses, and he just kind of put them together, and he's able to see these organisms through these lenses. And later on, Robert Hooke coined the term cell. And by the 1830s, the cell theory was proposed that all these living things had these small microscopic organisms in them. So all living things have cells. That's the first sort of like idea behind the cell theory, is that all living things are comprised of cells, and that life doesn't get smaller than the cell. So the most basic unit of life is the cell. Anything smaller than that does not qualify as life. Okay? And that we notice that new cells arise from existing cells. Now, we won't talk about much of this today, but we will in a few weeks where we go over mitosis. Okay? So that's what we're talking about today. All living things have cells. So let's talk about the super, super simple. Okay. We're going to, and I have a lot of Blackboard stuff to do today. So we're going to talk about cells. And I will start over here with prokaryotic cells. Okay. And then over here, I'm going to talk about eukaryotic cells. Okay. There we go. Prokaryotic cells, eukaryotic cells. What the heck are we talking about? Well, I like to break these words up. Prokaryotic, the first word, the first kind of I guess prefix of this word is procary or pro, which just means before in Latin. And then you have cary, which means nucleus. Okay. So you have pro, before, cary, nucleus. You put them together, it just means before nucleus. So really from the context, you're looking before nucleic cells with nucleuses are out. These are the cells that were here before there was a nucleus. They're simpler. So there's like an evolutionary component to them whenever you're naming these conventions. We have this naming convention. But the way we look at it is these have no nucleus. Specifically, no membrane bound nucleus. Um, so um, there's no nucleus in here. Whereas if you look at a eukaryotic cell, you break down that word. The first part is EU, which means true, and then carry, which is bacteria, or not bacteria, nucleus. Okay, so true nucleus, which means we have a nucleus, right? Okay, so when we're looking at the difference of prokaryotic versus eukaryotic cell, prokaryotic cells being more bacteria, your single-celled organisms, they don't have a membrane-bound nucleus. They have genetic material, but they don't have a nucleus. A eukaryotic cell has a nucleus that is membrane-bound, different from the rest of the cell. So it has a separation, a wall that separates the nucleus from the rest of the cell. So that's like the difference right there, the difference between the two cell types. Now, what do these cells actually have in common? Because we know what's different about them. So if I talk about all cells, okay, all cells, regardless of prokaryotic or eukaryotic cells, have four things in common. The first thing they have is a plasma membrane. And what do I mean by this? It means they have a way to keep the insides inside and the outsides outside. All right? They have something that separates the inside from the outside. Number two is you have cytoplasm, which is essentially the water of the cell, the aqueous environment of the cell, where the the genetic material, the organelles, they all kind of float in. It's that viscous liquid that keeps everything suspended and hydrated, and it's that barrier that allows the organelles to float around in. But yeah, that's the this aqueous environment inside of a cell. Um, very jelly-like, I guess is a good way to say it. The third thing that all cells have in common is DNA or some form of genetic material. And the last thing, again, DNA, what makes the thing the thing? So that's just the code. And the last thing that every cell has are ribosomes. And what the heck is a ribosome? Well, it's just a fancy way of saying this is where it synthesizes proteins. Okay? So ribosomes are the first organelle that I'm mentioning, and that's, where you synthesize proteins. But all cells have those in common. Go down further to a prokaryotic cell is just the prokaryotic cells do not have the membrane-bound nucleus, and the eukaryotic cells do. So Let me separate that right there. And what I'll do now is I'm going to actually draw my eukaryotic cells or my prokaryotic cell. So we're going to go here. Okay, I'm going to go. Mm, Okay, not the greatest, but you can, in fact, you know what? Can do that a little bit better. Okay, let's get my pen. All right. Okay, and I'll draw a little Jello right here. Doop, 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 doop. Get some little hairy guys sticking out there. Okay. And so then we'll just draw right in here, and then we'll come up. Okay. So we got this little cell right here, this little prokaryotic cell. Um, and I'm going to draw some genetic material right in here, and then I'll come down like so. Okay. So you got some genetic material. And to break this down for starters, we have this area right in here. I'm going to just highlight this in white right here so you can kind of see it. All right, this area where this DNA, this genetic material is called the nucleoid region. Okay? And then you have this genetic material, which is the DNA or the chromosome. Okay? So you have the DNA, the nucleoid region. Now, there's no membrane surrounding this cell, this nucleus. It's just kind of free floating DNA. All right. Um, Within these structures, you actually see floating around this nucleoid region these yellow guys. And remember, what were those things that all cells have in common? These are ribosomes. Okay. Now, ribosomes are very important in protein synthesis. They take genetic material, they make proteins. Okay. So these proteins are here. Um, there are these ribosomes here to help make proteins. And if you were to go even further here, so you have that within these structures, you even have this yellow guy is a cell wall. More specifically, it's called a peptidoglycan cell wall. The funny thing, or the interesting thing about the cell walls, these cell walls are what your penicillin attacks. So it helps rupture these cell walls so that the insides are no longer inside, they're outside. Okay? So you have a cell wall. You have a cell membrane here too, which I'm not going to draw, but it's in the same vein as the cell wall there. Housed inside the nucleoid region, you have the chromatin or the chromosome, the DNA with the ribosomes inside this nucleoid region. Now up here, this guy right here, we just call that a flagella, and this is for motility, movement. Okay, so that's a little swimmer guy. A good example of this it would be a sperm have a flagella to help get it up to fertilize the egg. Um, Also, what you have on here is you have these little guys right here. Okay? And these little guys are called pili. Pili. Okay? Pili. Um, so one thing that you have to do is that remember that DNA has this circular DNA. So it has this level of circular DNA. Um, and let me see if I can find some spot there. We call that a plasmid. Okay. So it has this unwound sort of DNA. the chromosome, but it also has this plasmid. Now the DNA, the chromosome DNA, is like it's bare bones stuff. And the plasmid is like learned DNA, like bacterial or antibacterial resistance. So this plasmid holds information that it's accrued over its lifetime from other bacteria. And how it transfers that information is through these things called pili. It can vertically or horizontally transfer um, it can horizontally transfer genetic material or genetic information such as antibacterial resistance. This is why they tell you whenever you're taking a medication, you finish the dosage. Because what can happen is you can leave one or two strands or colonies of bacteria that haven't been fully destroyed yet. They can get a mutation in them that allows them to be resistant to that bacteria. And then you're now dealing with something that can replicate and send that plasmid via the pili to other prokaryotic cells, other bacteria. And now um, that antibiotic does not work. So here's what yeah, so that is prokaryotic cells kind of in a gist. Um, and so when we're looking at this now, we're looking at this cell again. This is the most basic, bare bones cells. They're the prokaryotic cells. And they are the smallest of all the cells. Now, cell size matters. So if an organism is gonna grow, if it's gonna assert some dominance in this world, it needs to grow. And it can grow in one of two ways. It can grow in size or it can grow in number. Okay? Now, why is one better than the other? Now you have to do some math on this. Now, imagine that again, imagine bacteria and cells operate as spheres, as you can see here. All right. This requires a little bit of math, and I apologize from this. But anytime we're looking at a sphere, we look at or a three-dimensional object like a sphere, we're really concerned with the radius because there's two measurements that the radius is used in. It's used in surface area, which is the surface area of a cell is it's pi times the radius squared, and we look at volume, which is 4/3 times pi times the radius cubed. All right. So the ideal is that surface area and volume are relatively close to each other. So what do I mean by that? Let's just pretend I have one unit or one radius. We'll just say one radius. And if I do surface area and I do volume, okay, sorry, volume. And then over here we have 2R. So remember, if I did surface area and I have one radius and I square it, well, I have one. Okay. But if I have two, what's two squared? 4. Volume, what's 1 cubed? It's 1. But if I do volume, if I do a 2 cubed, I get 8. So in this particular case, I have a surface area to volume ratio of 1 to 1. And in this case, I have a 1 to 2. So as the cell gets bigger, there's more surface area or there's more volume you have to go across and less surface area that can cover it, if that makes sense. So what you end up having is you have to be able to transport nutrients across a cell. It's much easier to do it when the surface area and the volume match each other. But as I get bigger, it just becomes too energetically inefficient to maintain a cell that big. So how we've adapted, how life has adapted is we don't increase in size from a cell standpoint, we just increase in number. And it's because of this surface area to volume ratio. You want those to be as close as possible together. Okay? So, talked about prokaryotic cells. Let's talk about the eukaryotic cells. So, you go here. There's our prokaryotic cells. let's talk a little bit about our eukaryotic cells. Remember, okay? And what I'm going to do over on my last Blackboard slide is I'm going to title this guy Organelles, okay? Because I'm going to go over all those guys. I'm going to keep bouncing back and forth. So, eukaryotic cells. Eukaryotic cells are important because they contain that membrane-bound nucleus and they contain organelles. Um, so when I'm looking at this, let me make sure I got this right. So we're going to build a eukaryotic cell. I'm going to build it in a few different ways. I need to zoom in, zoom out. So I'm going to just go ahead and draw. Again, you have a nucleus and you have organelles. Those are your defining characteristics. Organelle just means little organ. Okay. All right. So taking my eukaryotic cell, all right, I'm going to kind of just build from scratch my eukaryotic cell. Kind of looks like an olive. It's not an olive. It is a eukaryotic cell. Okay. Inside this eukaryotic cell, this guy right here, this inner circle is called the nucleus. Inside the nucleus, we have a dense area of chromatin or chromosomes called the nucleolus. So up here we have the cell membrane. Okay. Right here we have the nuclear membrane. Okay. And here we have the nucleolus. Now, the nuclear membrane, the nucleolus, all that, that's the nucleus. Okay. Um, so the nuclear membrane is just that part, but this whole structure here is the nucleus. Okay. So that brings me to organelle number one, and these are all going to be in different colors, so I'm jumping back and forth, is the nucleus. And what the nucleus contains is genetic material in the form of DNA. Okay? This is what makes you you. You have DNA in you. Deoxyribonucleic acid. We'll touch more on DNA in a future week. But right now, you just need to know that nucleus is where the DNA is housed. Okay? This is system control. So we go back here. So this is where the nucleus is. All right? And then within that, you have this dense material. This is where the nucleolus is where the DNA is. And so nucleolus, DNA. You'll have these strands of what's called chromatin kind of floating around too, but that's also DNA. All of that's living inside of the nucleus. Okay. Um, and so, yeah, this is what you have to understand about the nucleus here or the cell. So this is going to get kind of not, I don't have a lot of space to work in here. So we're going to zoom in on the nucleus. We're going to work on the nucleus right here. Okay. So what I'm going to do is I'm going to just kind of make a big old nucleus. This is just the nucleus. And I'm going to build this nuclear membrane. Okay. There's your nuclear membrane. Now inside, we're going to get, that's not good at all, is it? In fact, I don't need to, here we go. We're going to build our nucleolus right here, just this dense material. Remember, nucleolus. Okay. Inside of this now, again, we have the chromatin kind of floating around. All right. This is chromatin, and it's just more genetic material that's floating around. Um, It's chromatin was just kind of DNA and some assorted proteins. All right. That's what these little lines are that you're seeing when you see these drawings. And you have the nucleolus, this condensed region of this dense region of DNA. And within that, so now here's the thing. Um, your uh your your nuclear membrane, your nuclear envelope, it is designed to keep stuff in, natively to keep stuff in, but it also has to get stuff out. So one thing you have to remember is that DNA, it stays in the nucleus. Okay? It stays in here. It does not leave the nucleus. All right? So the DNA stays in here. Okay? But I've got to get the genetic information, which is housed here, to the rest of the cell. All right? I've got to make it into something. Information is great, but if I can't make it into something, it's kind of useless. So DNA does not leave the cell. And in fact, if I were to, well, I'll just say I'll go in here, but RNA does. So we talked about DNA and RNA in the macromolecules talk. If I come up here, I draw my double-stranded DNA like so. Okay. So one of these is called the it's called the sense strand, and you have the anti-sense strand. We'll get into that later. But anyway, one of these strands can be used to code for RNA. And RNA can come out of these pores right here, or it can be coded onto a ribosome. Ugh. Okay. And that can help make proteins. Now, I'm going to explain that a little bit better. But you have the nucleus that has the nucleolus, the chromatin, the DNA that has all this information. It gets sent out from DNA, and more specifically, something called messenger RNA. There's also, so you have messenger RNA, you have ribosomal RNA, and you have transcription RNA. Okay? Or transcription RNA, tRNA. All these RNAs help code for protein that ribosomes use. Okay? So um, the ribosomes can help translate mRNA to proteins. Okay? So how does this happen? Again, this is where that nuclear bound membrane is, and this is why DNA and RNA have a little bit different nucleotides. Remember, DNA has A, T, C, and G, but RNA has A, U, C, and G. That U is what allows stuff to go out. It makes it just a little bit different. It keeps DNA in. So those hydrogen bonds within that particular sugar act a little differently, which the allows it to leave the nuclear membrane. Okay? So I have my RNA, I have my information. Now, one of two things can happen once I have my RNA. So let me go back to my final thing. So you have your nucleus, your DNA. Okay? Number two is I have ribosomes. Okay? Which is for protein synthesis. Okay? So ribosomes are good for protein synthesis. Again, DNA becomes RNA, and it could come one of three: mRNA, messenger RNA. It could become ribosomal RNA, which makes ribosomes. It could become translation RNA, which helps translate amino acids into proteins. Or it can just become, again, messenger RNA, which is just going to make maybe cholesterol or synthesize lipids. So everything's housed in there. The ribosomes help synthesize protein. And this is where again, this is the instructions that you get. So now going back to my cell here. Okay, there's your nuclear membrane. Let me get a different color. So attached to this guy is going to be like a. I don't know what I'm drawing there, but well, I do know what I'm drawing, but I'm kind of guessing here. Is you have, and I'll draw another one over here. Sorry for the the sounds there. Okay. This connective, these two parts right here collectively, and let me get a different one. I'll just use the same color right here. These guys right here are called the ER, the rough endoplasmic reticulum. So this is R, this is S. So you got this rough endoplasmic reticulum, which has covered in it these dots, ribosomes, hence why it's called the rough endoplasmic reticulum. And you have the smooth endoplasmic reticulum, which has no ribosomes. Okay? So we go back to this guy, connected on some of these little ports of entry here. And I'm going to draw it. Here we go. Let me see if I can. And I'm kind of making a giant endoplasmic reticulum right here. All these folds inside of it. This is where I can send RNA or ribosomes for protein synthesis. So here we have the rough ER. And again, I'm zooming in on the nucleus here. And then maybe over here, I would have something without the ribosomes. And this is the SER. Okay. And again, I would have ribosomes on these guys. So there's your rough, there's the roughness to the rough endoplasmic reticulum and the smooth endoplasmic reticulum. So I have my information. I have converted it to I've converted to I have something that can make proteins. But now once I've so the ribosomes make the proteins. Well, if I go to the RER, and I'll just trust that you remember that's the rough endoplasmic reticulum. Um, this is where I can do protein folding. Okay, remember proteins folded here. There's processing. Think of this entire mechanism as a big old factory. The nucleus has the DNA, has the blueprints. The ribosomes are the things that can assemble the proteins or put the amino acids together. And we'll talk about that process in a future week. But once they're together, the rough endoplasmic reticulum starts folding. Remember, uh, let's see, can I draw it here? Should I go back? So I'll draw it in the corner. Remember how you had that spaghetti monster that looked like a protein? Well, all that folding, again, you had the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. You had all that happening. Well, this is where happens. It happens in the rough endoplasmic reticulum. It folds it into the structure. Um, and it does this with sulfide bonds. It bonds two sulfurs together across chains to make these folds. So they form these disulfide bonds. And this is where this happens. And again, if it doesn't, if you have a misfolded protein or a misorganized protein, you can cause a lot of damage to your system if a protein's not folded properly. So the rough ER has a huge responsibility. Now, if I looked at the rough RER, I look now at the smooth endoplasmic reticulum, and what happens here? If proteins are in the rough endoplasmic reticulum, lipids and carbs um, are synthesized in the smooth endoplasmic reticulum. Okay, so this is where those monomers are put together. So here you could see your triglycerides, and your sugars. Okay, all those are put together here in the smooth endoplasmic reticulum. Um, And again, when you look at the smooth endoplasmic reticulum, this is a huge organelle that helps synthesize your cell membrane or helps maintain your cell membrane. Okay? So you have the smooth endoplasmic reticulum, you have the rough endoplasmic reticulum. Um, moving right along. So there was our nucleus, our smooth endoplasmic reticulum. I'm going to go back here and I'm going to label this. This is for proteins. This right here is for carbs, lipids. So your cholesterols, your steroids, all those things, your sterols are assembled here. So after this, going back, so you have the ER, all that stuff. Now you have to um, process and tag. So now you've made your protein, you've made your carb, you've made your lipid. Well, where the heck does it go? So the next thing you have to assemble here, or you have to look at, is this guy. And we just call this guy the Golgi apparatus. Okay? This is the Golgi apparatus. And this is where the you see processing, tagging, distribution of your stuff. So um, so I'm looking at the Golgi apparatus. So the Golgi apparatus has two facing spots right here. Um, and you have many Golgi apparatuses. So remember, whenever your proteins are folded, or your lipids are folded or whatever, it sends it to the Golgi apparatus. So that actually takes me to this guy, and we'll go down here, the Golgi apparatus. Okay. So we look at the Golgi apparatus. Again, this is distribution. Okay. So we distribute the stuff here. Is it going to help rebuild the cell? Am I going to send it out of the cell? Am I going to what organelle am I going to send it to? This is possible. This is the place where if a damaged protein is tagged for being damaged, then it's sent off to destruction or breakdown through the lysosomes or the peroxisomes. So the Golgi apparatus has a huge job. So you have two faces, and I'll go all the way back um, out here to the Golgi, and I'll draw the gold because eh, so you have two faces on the Golgi. You have you have over here on this side something called the cis face, and then over here you have the trans face. The cis face is what you get from the endoplasmic reticulum. So if it's a protein, whatever, it is sent to the Golgi apparatus through the cis side. Inside here is the medial, where it modifies the gold or it modifies the protein. It may add a sugar to it, it may add a sugar to it. It may add something to it to send it to a certain place. And then the transphase is what ships the protein or the cholesterol or the steroid or the lipid, whatever. So again, it does it through this process called glycosylation. It adds sugars to it. And however it's modified determines its function and destination. So an example like anything that's tagged with this is an example let's go over here. I'll write down here. So anything that's tagged there's a sugar uh there's a sugar that's called mannose 6 phosphate. If it's tagged with mannose 6 phosphate, it's sent to the lysosome. Okay? And the lysosome, again, that's where stuff gets destroyed. um but incorrect tagging can completely destroy the entire system. I mean, if you don't send the stuff to the right spot, you're having a bad day. And there's again a lot of genetic malformities that come from when you don't have a proper working Golgi. And so yeah, we're seeing that more and more. So now, again, that was the Golgi. Let's go all the way to the end here. Here we go. That was your Golgi. So you have information in the nucleus. Ribosomes are your factories that produce the proteins. But the RERs, the rough endoplasmic reticulum, fold it, uh, and they process it, and they tag it, and they send it to the Golgi. The smooth endoplasmic reticulum takes your triglycerols and your fatty acids, makes a lipid. Your glucoses, your monomers become a polymer, makes a sugar, it can fructose, glucose, all those things. Sends it to the Golgi. The Golgi distributes it. Okay, but that's the FedEx company, I guess, if I were describing the Golgi. But how do I transport these things? Well, I have these things. I have this transport system called vesicles. Okay, and vesicles are what carry the cargo in between. Are what carry the cargo in between stops. So I'm making these red dots as these vesicles. So it's picking up these vesicles and sending them along the way. But here's my problem. Okay, now I have transport, which is transport, but I need a cytoskeleton for my roads. Okay. And when I'm looking at the cytoskeleton, I'm looking at three different types. I'm looking at um, I'm looking at um, I'm looking at what you call microfilaments. Okay. I'm looking at intermediate filaments, and I'm looking at microtubules. Okay. What's the difference between all of these? Okay, the microfilaments, let's go back and how do I want to do this? I'll just go back here. So you have the microfilaments here. You have the microfilaments here well, we'll start with the intermediate filaments. So these intermediate filaments kind of like, I'm just going to draw X's at the edge here. Okay, kind of floating around up here, or actually, they're providing a lot of structural support. They're what makes sure that the cell keeps its shape, and if there's any pressure, the thing doesn't collapse in on itself. These are what's called these little X's here are the intermediate filaments. So the IMF, the impossible mission force, if you're a Mission Impossible guy. But that's the IMF. The intermediate filaments are happening right there. Um, so then you have like the microfilaments, uh, which help with flexibility and movement. So if cells need to move a lot to flex a lot, they have a lot of microfilaments. So let me get some blue here, and you may see them kind of like so. These are your microfilaments. Your microfilaments. So microfilaments are usually composed of something called actin and myosin. These are big in your muscle cells. So contracting, actin and myosin, those microfilaments help move and flex the muscles. The intermediate filaments make sure that when you're flexing, they don't break something. They provide that structure. But um, within that, you have something called microtubules, and microtubules are kind of nice because they're very big with transport. So I'll come over here and they kind of have this globular like shape to them. They're kind of like these cylindrical tubes. And if I imagine I drew those all the way up. And so you have this tube-like structure that is again found in here that allows transport that the vesicles can transport through. So these vesicles come straight through there, taking whatever they need to take wherever they need to take it. And this is the cytoskeleton. This is really cool. So if we look at the cytoskeleton, it has these roads and this tensile strength that acts as bridges and flexibility and motion, but also transport within the cell from organelle to organelle. So it may transport from the ER to the Golgi, from the Golgi to the lysosome, from the lysosome to the cell membrane. Wherever it's being tagged, there is you got to get it to where you need to get it. So what's next? What is next? Well, okay, that's great. We have our cars, we have our roads, we need some gas to get the cars going, right? Well, this is where your mitochondria come in. And I think anybody who's taken any intro to biology class knows that the mitochondria is where ATP synthesis is, which is where you get your cell energy. And you get that from the breakdown of glucose. Okay? That's C6H12O6 broken down. You get adenosine triphosphate. And that is your energy, mitochondria, going back to our cell. Let me get some purple here. And these are what these guys are, these little mitochondria. They kind of draw them like that. These little cristae formations, these internal foldings that increase surface area, are your mitochondria. And this is where you produce ATP, which is your energy, for your cell. Okay. Um, Now in we're drawing a eukaryotic cell right now. All right, we're drawing a eukaryotic cell, which is an animal cell, but plant cells um, have another organelle, and we will actually kind of just draw. And if I'll go over to my list here because you have mitochondria and but also in plant cells, you have the chloroplast. So, and the chloroplast is the site of photosynthesis. So, we will talk about photosynthesis in detail later. Uh, but this is where plants take sun, make glucose for their own energy, whereas we have to eat our food to get our glucose. They can make it through the sun. Plants also have mitochondria, but the chloroplast is very important. Um, So yeah, so now we have energy systems within this. So these guys right here, energy. These guys are like transport. Okay. And I'll break this down here. I don't want to get this in 7, 8, 9. And let me get another color. Get some white here. And then we're looking at lysosomes and peroxisomes. Okay. And what are we dealing with here? We're dealing with breakdown and protection. And I'll touch on that more in here in a minute. Um, you also have vacuoles, and that's for storage. Got to store this stuff. And then you've got to be able to communicate between cells. And I'll touch more on all this stuff here in a second. So, okay. So we talked about the chloroplast, lysosomes, vacuoles. You have to communicate too. So let me go back here. All right. So here we're going to talk a little bit about lysosomes and peroxisomes. Okay. So what are we talking about when we talk about lysosomes and peroxisomes? Okay. Well, lysosomes and peroxisomes are just simply ways that our system maintains the junk. What do I mean by that? Um, so remember we talked about lysosomes, or we talked about hydrolysis, lysis, breaking something up. Lysosomes are very big, and we'll talk about lysosomes with pathogens. In fact, your white blood cells have an abundance of lysosomes in them because they help with dealing with pathogens. They sort of I guess the way to look at it is they will sort of ingest your pathogens and just destroy them. They're a very acidic environment. Remember that. These are all around your organelles. And again, it's for protection against pathogens. They also are good with old organelles, like organelles that uh, have a worn out. They're not being used anymore. That's where you recycle them. Um, Peroxisomes are good for detoxification. They are good for handling these things called free radicals. Okay, what is a free radical? Okay, so now you may have heard this terminology. Well, peroxisomes are pretty nice because they handle these things called free radicals. The biggest one is oxygen and unbound oxygen. So remember how I said oxygen with its six valence electrons likes to be bonded to another oxygen that has six valence electrons. That way they can share two. There's that octet rule. Okay. But sometimes oxygen is a free radical. It's kind of binding around. It has its six valence electrons and it wants two more. So it will steal two electrons from other things like maybe your bones or your organs. These is free radicals. Peroxisomes help neutralize this guy. They'll absorb the oxygen and they produce something called H2O2, which is peroxide. There's your peroxisomes. Think of hydrogen peroxide. And then they break that down to H2O and oxygen. There's that O2. So when we talk about free radicals, the idea is that it's going to steal electrons from a neighboring organelle, a neighboring structure, and we're trying to stop that from happening. Peroxisomes help with that. Um, and then more so, we talked about again lysosomes and peroxisomes. A lot of cells have these giant storage units called vacuoles. Okay. Um, they store water, nutrients, waste. In plants, they maintain pressure. They push out on the cell wall. Um, and that is something that we haven't really talked about is the difference in the plant cells. So I will kind of dovetail and I will talk about, well, I'll talk about the two competing things between plant and animal cells. Um, but there's our eukaryotic cells. And again, eukaryotic cells, this is more animal. Okay. If I were to go, say, look at like plant cells, plant versus animal. Okay. So in this particular case, you're looking at this is your animal cell, kind of very circular in nature, whereas plant cells have that more rigid structure. Um, and this is because in this case you deal with a cell membrane, and in this case you deal with a cell wall. Okay, so that's your plant versus animal, your big thing. The other big thing is if you're going, is you have in here, and again, I'm just writing these out. You'll have chloroplasts in here, again, for photosynthesis. Okay, and you'll have very large vacuoles for storage, and that helps push on the cell wall. These guys do, okay? They push on the cell wall, kind of giving that rigidity. You do not see that. You see smaller vacuoles here, um, and no chloroplast. Okay. So going into the individual differences within these two. So going right along. Okay, so the next thing that I want to talk about, again, going into these differences, plant versus animal cells, which I'm going to say specifically, I'm going to do a cell wall versus cell membrane. Okay. Okay. And then I will do a cell wall down here, and we're going to do a cell membrane down here. Okay. Cell wall, cell membrane. We'll start with the cell membrane because this is something that's, I mean, I've talked about both in general. Um, but cell membranes uh, are composed of two items here. And I'll kind of just draw this down here. This is our cell membrane. Let me just go circle, 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 circle. Okay. Circle, circle, circle. And I'm drawing these two legs on these guys. And I'll zoom into it so you see. This is essentially your cell membrane right here. Okay? Now, if I look at the anatomy of a cell membrane, again, so just pretend this is the outside of the cell. This is the inside. Okay? How do I keep the outside outside of the inside inside? Well, this is what's called the phospholipid bilayer. And so I'm kind of zooming in on like this guy right here. Just pretend I'm zooming in on that guy. This top guy is a phosphate group. Okay. It's a phosphate group which loves water, hydrophilic. And then these fatty acid tails are hydrophobic. So they don't love water, right? So what ends up happening is in a cell wall or in a cell membrane, these phosphate groups, these phosphate heads, orient themselves to point towards the aqueous environment. In this case, the cytosol or the plasma outside of the cell, the water environment outside of the cell, so to speak. Um, the phosphate orients that, but because of these fatty acid tails right here, H2O or the junk on the outside cannot get in and it cannot get out. Okay? So when you're looking at a cell membrane, you're looking at a phospholipid bilayer. Okay. That is a cell membrane. Your book talks a little bit about this. It talks a lot about it. It's a very big thing. Um, but the phospholipid bilayer is what allows the insides to stay inside and the outsides to stay outside. If you want to get more specific, when we talk about the cell membrane, we'll have a whole chapter dedicated to this. You have transport units that are able to send stuff across the cell membrane. It's how you get stuff in, you get stuff out. But the cell membrane is composed, and this is what animal cells are, what you and I have, a cell membrane. They have this phospholipid bilayer. They have a phosphate group attached to a tail, and those tails orient themselves inward away from the water. Okay? So now if you're looking at plant cells, plant cells have a cell wall. They don't have this phospholipid bilayer. The plant cells, again, they have that square, that rigid square on the outside here. And again, I just kind of visualize them better as squares because they're more rigid. But the cell wall um, has, again, it's a wall, duh, that's made of cellulose. And what is a cell wall that's made of cellulose? Well, remember, cellulose is just a polymer. It's a sugar polymer, I should say, and it's just a lot of glucose or simple sugars bound together. Now, how are they bound together? Now, remember that glucose has that's a bad hexagon. Glucose has is a six carbon sugar that has that ring-like structure. There we go. And all right. And remember, like if I have yeah, it's these when I have the glucose molecules bound in this orientation and I keep going out like this. So you have these beta linkages. Remember that? Those beta 1,4 linkages because of the different orientations of these oxygens, how they flip. Whereas if they were on the same side, they'd be alpha, and that would be your starch, your glycogen. But in plants, they orient themselves this way to make the cell wall. These cell walls are very hard to break down. Cellulose. This is the difference between the cell wall and the cell membrane. One of the big things I ask this every year, is I say, hey, why are cell walls, or what's the difference between the cell wall, or what's the difference between cellulose and glycogen? And it's the beta 1,4 linkages and the alpha 1,4 linkages. Okay. Um, so yeah, that is the generic difference between the cell wall and the cell membrane. Now, if I want to look a little further, make sure that I have everything. Come here. Here's all of your organelles. The last thing that you want to talk about when you're talking about this stuff, we've talked about the generic functions of all these organisms. We've talked, or these organelles. The last thing to really talk about, and again, a lot of times, um, when I see these lectures done, you stop at the organelles and internal transport, internal cellular communication. But you really don't talk about how cells are connected. Um, and you have to the cells have to talk to each other. If one cell's failing, it needs to tell that to another cell. It needs to be able to send nutrients to another cell. So going there we go. There's our cell wall, our cell membrane. But how are cells connected? Okay, we'll just go over here and just try this again. How are cells connected? Okay, are they connected? So if I have, we'll just take some animal cells here and we're putting them together like so. Maybe these are epithelial cells, or, maybe they're bone cells or they're white blood cells or whatever. But these are individual cells. So, and more specifically, they're animal cells. And if we really wanted to do plant cells, we could do, and these are plant cells. Okay. So you're looking at these connections. So how do I get information from here to here, here to here, and so on and so forth? Okay. Um, you basically have to have there's three types. They're called intracellular junctions. Okay. And one of the ones, and we'll start with plant cells. Um, plant cells are very thick. Plant cells have something, and I will label this. Well, how do I want to? In fact, you know what? I'll just draw a line through here. I probably should have done this differently. Plant cells have something called plasmodesmata. Okay, and plasmodesmata is just there's just structural modifications. They're just channels that allow nutrients, ions to pass through channels through the cell wall. I should just put through cell wall. Okay. These are plasmodesmata. They kind of have little channels right here where the information can kind of just pass right through there and there. Okay. So that's plasmodesmata. That's essentially there's just little junctions within the cell walls that they're connected together. Then they can pass through adjacent cells on through the plant walls or plants cell walls. Um, Now animal cells have a little bit different mechanisms. Again, plasmodesmata is the big one that's in animal or in plant cells. But you're looking at within animal cells, you're looking at things called gap junctions. You're looking at tight junctions. And the third thing you're looking at are desmosomes. So that word should look very familiar to you. Um, so these gap junctions right here, if plasmodesmata are those channels from animal cells or plant cells, the plasmodesmata or the gap junctions, sorry, are those channels between animal cells. And they're just direct channels. They're like rapid communication between cells. So this is like the primary one that you see. Um, these tight junctions right here, again, you don't want a bunch of information leaking, but these tight junctions are kind of like spot welds that make sure that these things stick together. And then the last thing that you have is well, let me rephrase that. The desmosomes are more like the spot welds that produce a lot of resistance, a lot of anchoring under stress. And then the tight junctions are more of they more seal things together, like a watertight seal. Um, but the communication is gap junctions, and the plasmodesmata is the communication through the cell walls. But tight junctions and the desmosomes operate well, tight junctions operate in both, and desmosomes are just plant or just animal cells. I'm probably very confusing right now. These are animal cells. These are plant cells. Um, Gap junctions, tight junctions, these operate in both. So to sum that up, we'll go back to plant cells, which has the plasmodesmata, which are those channels in between the cell wall. Plasmodesmata. Okay. Animal cells have desmosomes, and these are just channels that share information. Okay. Now, both of these have the tight junctions and gap junctions. Okay. Which are basically how the cells stay connected. Golly. All right. And that kind of wraps everything up. So let me bring it all home with one conclusion. Let me get a different color. So, in conclusion, what do you need to know? You need to know the organelles. Okay. You can go back and you can do this all again. You can look at all this again. You have the nucleus, you have the endoplasmic reticulum, Golgi, ribosomes, cytoskeleton, mitochondria, lysosomes. Each step depending upon the next. So organelles, you need to know form and function. Okay. Form. I cannot write today. Organelles form and function. I will ask you, like I could ask you a question, if a lysosome fails, what could be the next metabolic process that happens? If I have a you'd have to know that if a lysosome fails, you're having a buildup probably of pathogens in your system, so you might be getting more sick. You have to think about that. Uh, what if my mitochondria fails? No energy. what if protein foldings fail? Or I should say, what if the endoplasmic reticulum fails? Well, then the protein folding, I get misfolded proteins, which again can cause, autoimmune disease, detrimental things like that. So yeah, just knowing the steps, organizing them, is going to be paramount. So again, I'll write it out here so you can have it. So you have the nucleus, which has information. It's got to send DNA to RNA to the ribosomes, which makes protein. Okay. Protein, and I'll just loop this back around. Protein is then folded in the rough ER. We won't exclude the smooth ER because it's making cholesterol and lipids. Okay. The Golgi apparatus. Okay. Then tags and sends it. Um, the vesicles transport it. The cytoskeleton, which are the microtubules specifically, help transport that. And anything that's not used, that could be in the cell, out of the cell, and anything that's not used goes to the lysosomes and the peroxisomes. Okay. But understand all that. You connect them all through junctions too. This is how tissues and organs form. So an individual cell comes together, those junctions make bigger systems. Okay? So that's kind of the nutshell what you need to know. Um, I think that's it. If I forgot something, I will send it in a message. I will send it in a text, or not a text. I'm not gonna text y'all. I'll send it in an email. That being said, this is Chaz Stevens signing off. And what do I always say? Stay curious. Have a great day.